Wait! Wait! In the last 5 seconds, over 5 million cells in your body have died. That's about 1 million cells a second. But don't mourn yet. The reason we aren't wasting away every few minutes is that another 1 million cells replace those every time they die. In fact, in the human body, there are over 100 trillion cells. That's 100 trillion. That's a huge number! Do you know how big 1 million is, let alone 100 trillion? If you were to stack 1 million 5-foot-tall people on top of each other, they would reach about 947 miles high. That's approximately 172 times the height of Mount Everest. And about 102 times as tall as the highest clouds in the sky. If you were, however, to stack 100 trillion people on top of each other, their distance would reach about 20 times the distance between Earth and Pluto. These are crazy huge numbers we're dealing with. But why do 1 million cells die every second just to be replaced? Well, I'm going to tell you in just a second. Programmed cell death is a process that's been around for over 1 billion years. That's so long ago that there weren't even multicellular organisms. The only life on Earth was bacteria swimming around in the ocean. So why has this process been around for so long? Well, cell death can occur for a number of reasons. Sometimes cells die because they are hurt. Ow. Cells can also die due to physical or chemical stress. A lot of chemicals made by the body can lead to cell death, but also harsh sunlight or radiation can cause DNA damage, which can trigger cell death.
Too bright.
Infection can also lead to cell death. When a pathogen, or something that causes a disease, enters a cell, the cell will either kill itself or another cell will kill it. Sometimes cells simply aren't needed anymore, and they become superfluous. So, now we know why cells die, but how do they die? There are 3 main types of cell death. There are more, but they're more specific and less well studied, so for now we'll focus on the 3 big ones: apoptosis, autophagy, and necrosis. The first type of cell death is apoptosis. All of the contents of the cell remain in their prepackaged membrane, while the nucleus of the cell shrinks around it. The plasma membrane, or outer layer of the cell, then blebs, which is an actual scientific term, which means bubbles and contorts. Finally, all of the DNA of the cell are chopped into tiny little pieces, and it looks something like this. The second type of cell death is autophagy, or literally self-eating. Basically, what happens is a cell creates little digestive packets, or vacuoles, which eat all of the cell's contents. This usually happens when apoptosis is blocked for some reason. The third type of cell death is called necrosis. This is the messy one. In necrosis, cells swell, burst, and then decompose. Okay, so there you have it. The 3 types of cell death: apoptosis, autophagy, and necrosis. So why do we care about cell death? Well, actually, during development, a lot of limbs and organs shrink due to apoptosis. If it weren't for cell death, we'd probably still have tails. And we'd also still have webs on our hands like a frog or a fish with fingers. Also, some of our forebrain cells die. If it weren't for cell death, we'd probably look something like this. Another important aspect of cell death is what happens when it goes wrong. Now, you've probably heard of cancer. Cancer is a very serious illness in which cells don't die because of a mutation in their DNA. Tumor cells just keep growing and dividing until they form a giant lump inside your body, which can make you really, really sick. Now, all of this happens because of a problem with, you guessed it, cell death. Now, we already know that cancers can happen because of a mutation in your DNA. But the mutation rate of DNA is about 1 in a million. And if we're creating 1 million new cells every second, then it seems like we should get cancers every few seconds. So why don't we? Tumor suppressors. Tumor suppressors are transcription factors, or proteins that read DNA and search for mutations. They instruct cells to die whenever they find a mutation by, you guessed it, apoptosis. The most important tumor suppressor is p53. But sometimes there's a mutation with the gene that codes for p53, and still other cells don't have the gene at all. And that's where things get pretty bad. But don't get too worried. There are so many checks and balances in place that cancers are really, really rare. Unless, of course, you like to hang around sources of gamma radiation. Now, I could tell you a lot about the basics of cell death, but it'd be really exciting to go and talk to someone who's actually doing research in the field. So let's go and talk to Doug Green of the St. Jude Children's Hospital and see what's actually currently happening in the field of cell death research.
Our lab studies how cells die and how they stay alive. We study it in the context of cancer and the immune system, but also in other circumstances. We study the molecules that are involved in cell death, what happens as the cells die, and the consequences for the body. We use a wide variety of techniques to study cell death, but among the most important, is single-cell biochemistry. It's watching cells with imaging techniques and exploring how the molecules within that cell are interacting to produce the process, in this case, the death of the cell. Why is it important to study cell death? We're like Goldilocks. We can't have too much or too little. It has to be just right. When we have too much cell death, we can have deterioration, degenerative disease, such as occurs in some cases in aging. Or if we have too little, we get accumulation of tissue, and that occurs during cancer. Understanding exactly how cell death occurs and how we can control it helps us control this process in the body. In the last 25 years that I've been studying cell death, I've seen the process go from a phenomenon, something that happens and looked interesting, to something that we're understanding at a fundamental biochemical and molecular level. Along the way, drugs have been developed that allow us to intervene in the process, and those drugs are in clinical trials in cancers with remarkable effects. The study of this process is so fundamental to biology that, of course, it's impacted on many different fields. And there's still a lot more to do. For me, the coolest thing about cell death is the fact that cells don't just die because they're injured or hurt. They actually kill themselves. They participate in the process of cell death. And that's very important for the consequences of that death for the physiology of the body.
All right, so let's review what we've learned. We know that every second, 1 million of our 100 trillion cells die. They die due to physical or chemical stress, to injury, infection, or just superfluity. There are 3 main types of cell death: apoptosis, autophagy, and necrosis. Cell death is an important factor in processes like development and in diseases like cancer. And all of this is happening in less than a second every second. Now that's what I call science in a second.